Hello everyone, welcome back to a new video. So today we'll be discussing about the platelet. I've divided this video into two parts. The first part will be consisting of the basic structural integrity or the complexity of the platelets. So without any delay, let's get started. Firstly, starting with the introduction, what does platelet means? So it has been said that platelets, they are also known as the thrombocytes, are the formed elements of blood. We all know that the blood mainly consists of the three major components, the RBCs, WBCs, and the thrombocytes or the platelets, and platelets being one of them. And they are mainly responsible for the coagulation of the blood. And also platelets are small, colorless, non-nucleated, that is they don't have any nucleus, and moderately refractive bodies. Next, coming to the size and shape. So if we look into the size, the diameter ranges from 2 to 4 m with an average of 2.5 m, and the volume ranges from 7.5 cum, that is cubic per micrometer, to and on an average, and it ranges from 7 to 8 cubic per micrometer. If we talk about the shape, so platelets does not have any definite single shape. They have varied shape according to various circumstances, such as it can be spherical or rod shaped, or it can even be oval or disc shaped when it is inactivated. However, some other peculiar kind of shapes also include the dumbbell shape, the comma shaped, a cigar shaped, or any other unusual shapes. Next, the most important thing, the structure and the composition. So, platelets is constituted by mainly the three components. Firstly, the cell membrane, which is also known as the surface membrane. Next, we have the microtubules and the cytoplasm. So, first starting with the cell membrane. The general description of the cell membrane is that it is 6 nanometer thick, and extensive invagination of cell membrane forms an open canalicular system. And this canalicular system is a delicate tunnel system through which the platelet granules extrude their contents. Before explaining this canalicular system, I'd like to show you the diagram about how it really looks. So this is the structure of the platelet when it is seen through an electron microscope. So if we look closely into the diagram, we can see that here, this blackish line, we have the cell membrane over here, and this invagination of inside the cell of the cell membrane is nothing but what is known as the open canalicular system. So this is what we were talking about. That is the cell membrane forms an open canalicular system which invaginates towards itself, and it is also by through which the platelet granules extrude their contents. Means via this canalicular system, the platelet throws away its waste material outside the body and other metabolites. Next, coming to the cell membrane. We all know that cell membrane mainly consists of lipids. So same is here. It contains lipids in the form of phospholipids, the cholesterol, and glycolipids. Also carbohydrates such as glycocalyx and glycoproteins, and along with that it also has some additional proteins. So this is the structure of the cell membrane and the composition. And last but not the least, we have the microtubules. So microtubules, again, if we see into the diagram, we can see here that this red dot structure inside the cell, that is in the cytoplasm, are nothing but the microtubules. So the microtubules form a ring around the cytoplasm below the cell membrane, and also they are made up of polymerized proteins called tubulin. This is important, the tubulin. These tubules, what is their main function? Provide structural support for the inactivated platelets to maintain the disc-like shape. So the main thing is that this microtubule contains a protein known as the tubulin, and they are responsible for maintaining the disc shape of the platelet only when they are inactivated form. Next, the basic and the most important component, that is the cytoplasm. So the cytoplasm of platelets contains the cellular organelles such as Golgi apparatus, endoplasmic reticulum, mitochondria, microtubule, microvessels, filaments, and granules. Along with that, it has some other chemical substances such as the proteins, enzymes, hormonal substances, etc. Next, if we look into the composition of the cytoplasm, then it contains various proteins such as the contractile protein. Now among the contractile protein, it has again various kind of subdivisions such as the actin and the myosin and other. Next, we have the von Willebrand factor. Now, this von Willebrand factor is mainly responsible for the adhesion of the platelets to the injured site. Next, the PAF, that is the platelet activating factor. From the name itself, it is suggesting that it is activating the platelet whenever it is required. Next, we have the PGDF, which is also known as the platelet growth derived factor. Now, the main role of this PGDF is that it helps in the formation or the repairing of the blood vessels. Next, we have vitronectin, which is again another protein that is responsible for the adherence of the platelets to the site, and also the last but not the least, the platelet stabilizing factor. And this is a very important because the platelet stabilizing factor actually prevents the formation of new blood vessels at the site of the injury. And again, it also, along with the proteins, it has other components such as enzymes, for example the ATPase, and hormones such as the 5-HTs, adrenaline, etc. Now, if we look into the normal count and variation, it is both very important in both physiological and pathological point of view. So, generally, it ranges from 2 lakh to 4 lakh per cubic mm blood, with an average of 2.5 lakh per cubic mm blood. Any derivation or deviation from this range will be considered as a physiological or a pathological abnormality. So, if we look into the variations, it depends on age, sex, altitude, and also other factors. For example, if we look into the age factor, then the number of platelet count is generally less in infants, but however, it reaches the normal level by the 3rd month. If we talk about the gender, then it is equal in both male and female. However, in female, it decreases slightly because of the menstruation. If we talk about the altitude, then it slightly decreases in high altitude, and also another factor that after intake of food, it slightly increases. So this is about the basic structure and component of the platelet. In next video, we'll be talking about the physiological functions of the platelets in detail. If you have any confusion regarding this video, do let me know down in the comment section below. Thank you.